Okay, excuse me, going to recording. And she was the leader for many scientific expeditions in Brazil, I noticed from her CV, which was really cool, for both Louisiana State University and for the Universidad de Sao Paulo between 2009 and 2019, and has participated in lots of other expeditions as well. Anna Hiller is also an ornithologist at LSU, working on her PhD with Brant Faircloth and Rob Brumfield. Anna received her bachelor's in environmental science from UC Berkeley in 2014, working with Drs. Carla Cicero, Rory Bowie, and Rosemary Gillespie. In addition to her scientific work, Ms. Hiller has spent time conducting teaching and outreach, especially working on empowering women and girls to enter the sciences. Again, I noted from her CV, that's wonderful work and so necessary. She has worked in Peru and Bolivia, among other locations. So please join me in welcoming our two speakers tonight and talking about their important work in the Amazon. Take it away.
Thank you. Thank you so much for this kind introduction. Yeah, I feel so much more important than I actually am after this introduction. So yeah, thank you so much. Thank you so much for everybody that came to watch this talk. And it's a very special talk for us. It's about this expedition, the Emilie Snethlage expedition. Unfortunately, tonight I cannot spend a lot of time with you guys, but Anna will take over and you will be in good hands. So I'll just start. We start with introductions, but Linnea made a much better job than I would do.
Do them anyway.
And what is special about this expedition is that I had this idea to let this team of 8 women biologists, ornithologists to the Amazon forest. And this idea starts because here at the LSU Museum where we work, we have a ton of role models. We have a ton of LSU alumni that inspired us and our job, especially in neotropical ornithology. And here you can see some pictures of them. And we have the pictures of these people that passed through LSU, either doing their PhDs or their masters here. And we are talking about like Dan Lane, Joe Craycraft, John Bates, Van Ramsey, that was the previous bird curator here at LSU, and many others like Ted Parker, that was considered the best neotropical ornithologist of all times, that really are inspirations for us and for the work that we do here, trying to describe the diversity and understand the evolutionary processes behind the formation of this amazing diversity in the neotropics, or in other words, in Central and South America. So these people were all role models for us, but what you can notice is that they are mostly or almost, totally just men. And that came to my attention when I was just starting my PhD here. And I started thinking what I was doing to change the scenario or to bring more diversity to museum field and museum ornithology. And I noticed that I wasn't doing like basically anything, I was doing a ton of expeditions and surrounding myself with all my male friends and colleagues, and doing these expeditions without thinking about diversity and without thinking about the need of providing more opportunities for women in these expeditions. And in many expeditions, I was left out just because I was a woman. So that's when I started thinking about all women expedition, and I thought that that would be a good opportunity to pay a tribute to the most important of my role models. That was Emilie Snethlage. So, and I wanted to make Emilie Snethlage more known to North American ornithologists and birders, because in Brazil, or for Brazilian ornithologists, she is quite famous. So Emilie Snethlage, how she was known in Brazil, she was this German naturalist, one of the first women in Europe to get a PhD. And she came to Brazil in 1909 to work as a naturalist and to be the bird curator at the Getty Museum in Brazil. And I first learned about her by reading the Bible of the Brazilian ornithology, this book that was written by Helmut Sick. And he dedicates his book to Dr. Emilie Snethlage. And what caught my attention when I first came across this page of the book was that Emilie Snethlage was missing a finger. When you look for the history of that, I discovered that in one of her expeditions to the Amazon forest, she was just playing in the water with her hands, and then a piranha just came and ate a piece of her finger. And then she was in the field and she had this infection, and then she felt that she needed like to amputate the rest of the finger to be able to keep doing field work. And then she started asking around in the camp, okay, could anyone come and just cut the rest of my finger so I don't have this infection and I can keep doing field work? And nobody wanted to do it for her. So she got a machete and she cut the rest of her finger herself. And this is the kind of role model that I want for me, you know? That's
Go girl.
that is just like a joke about this amazing woman that would go and do these expeditions by herself, and in times that she was, you know, she didn't want to shock people where she was going, so she would use like her skirts to go to the field, and would keep her long hair so she wouldn't like shock people where she would go. And she was just doing these expeditions in all parts of the Amazon forest. And in her career, doing this part of field work and museum ornithology, she described around 60 bird taxa, new bird taxa to science. She produced 40 articles in perfect Portuguese, German, and English. She came up with the catalog of Amazonian birds. That is still a big reference for any of us studying Amazonian birds. And she collected and preserved specimens in museums, like thousands of specimens that are still used today. And they are mostly housed in Brazil and in Germany. And I can only imagine what kinds of barriers and things that she had to overcome in that time to be a field and museum ornithologist, to be like the first woman head of a scientific institution, because she became the director of the Museu Paraense Emílio Guilde in Brazil. And I can only imagine like the challenges that she had to overcome. And because we are still facing these kinds of challenges. Because when I would tell people about this expedition, that would be some sentences, some expressions that I heard from people that I really admire, and people that are professors and that are important people in institutions, like thinking that having an expedition, an all-women expedition, in an isolated place in the Amazon forest wouldn't be a good idea. And that were just some things that I heard, but most of people were really supportive. But still, I think that when people would think of an expedition, an all-women expedition, that would be the first thing that would come to their minds. But in reality, for a lot of the expedition, what was really the case here, and to make an expedition happen, most of the time was spent by us like this, because the most difficult part to come up with an expedition in the Amazon forest, an expedition to collect specimens for our museums, is dealing with the bureaucracy. And I was lucky because in this process, I had my friends supporting and helping me. And this all starts with me sending this email to my friends, Jessie Salter, Anna Hiller, that were in my cohort here as PhD students in the museum by the time. Anna is still here, Jessie already graduated. And from now on, now that I told you how I got this inspiration for this expedition, Anna will tell you how the expedition went and what were the results for this expedition. And I will leave you guys with her, and I will take off just to keep doing my dissertation and try to get my PhD, because my defense is in the next month. So go ahead, Anna.
Yeah.
Yeah, no problem.
Thank you for the overview. Thank you.
Glaucia recently got a job, so she has to go defend to then start that job. So that's what we're gonna go let her do. But
yes,
I will take it from here.
Bye.
Okay. Thanks, Glaucia.
Thank you.
Thank you.
Yeah, so Glaucia sent me that email and I immediately responded like, yes, I'm so excited. I think this is a fantastic idea. In large part because I myself, these are photos of me on expeditions to Peru, to Bolivia. And it's, as you can see, like, again, a bunch of dudes and me. So I was so excited to get to go on my first expedition. This was the first expedition I got to go on that was majority women. And it was a really wonderful opportunity. But to make that happen, especially, I'm not Brazilian. And even though Glaucia was leading the expedition as a Brazilian researcher, there's many steps you have to take to be able to bring foreigners into Brazil so that they're legally like allowed to do research. So first we had to write, and by we, I mean mostly Glaucia, had to write what's called a scientific project and propose like, where do we want to go and what do we want to study on birds? And then we had to get expedition permits, so permission from the protected areas we wanted to visit to go collect birds. Collecting of birds is like very highly regulated, as you can imagine. And then we had to do something called the publication of names. And this is where Glaucia, who is the expert at navigating Brazilian permit bureaucracy, had to get all of the names of all of the foreigners going on the expedition published in an official government document by the country of Brazil, so that we could get what's called a research visa, which would allow us to go officially on this expedition. So it was going great. Glaucia submitted the project in March of 2017, and then it was accepted in February of 2018. And in March, she got a grant to help fund the expedition, which, as you can imagine, is a very important piece of any large, expensive trip. And then in February of 2019, the permit was accepted. And we were waiting and waiting and waiting. In the meantime, we were able to do some things like this is a wilderness first responders course. So big expedition, especially remote, you want to have everyone well trained in like not just first aid, but really emergency response medicine. So we did this. It was like a week-long course at the LSU gym. It was really fun, learned a lot. And this is me with some nice fake blood on my leg as part of the course. But we were still waiting on our research visas. Finally, in I think it was 1st of August, here's the date, 1st of August of 2019. So the expedition was like supposed to start, I think, August 6th. We ended up having to delay the start of the expedition because we still had not received this document. But we finally got it on the 1st of August, 2019. After much, much hard work by Glaucia, 86 emails, 57 phone calls, many, many prayers and stressful meetings later. This is a photo on the 8th of August, 2019. Half the expedition, all the Brazilians were already in Brazil on the boat waiting for us. And Jessie and I drove to Houston, not once, but twice, But the second time we successfully got our Brazilian research visas. So here we're very happy and sent this photo to Glaucia, who said it was the most relieved she'd ever felt in her entire life. There we go. That was the feeling. Yeah, so then we got to go to Brazil. From all over the US, we had researchers flying in from Albuquerque, Baton Rouge, which included myself, other researchers from Manaus, Recife, and Sao Paulo, everybody flying in to Caruarí, which is this very tiny town in the middle of the Western Amazon and very, very difficult to get to, which is part of the reason we were interested in going there to research the birds. Glaucia and her partner Marco were in charge of getting all the gear there by themselves, including 3 very large heavy nitrogen tanks, all of the equipment to prepare specimens. But it was a very cushy boat ride, so that was, I think, quite fun. I was in charge of getting sort of the American half of the team there, and we, well, along with a couple of the Brazilian colleagues, flew on this like tiny propeller plane, which went great, but Glaucia had forgotten to tell us that 1 month preceding this trip, that exact plane going to Caruarí had like crash landed. She told us this when we arrived on the tarmac safely. They're like, oh yeah, I was glad to see you because, you know, I didn't want to tell you this, but we'd had problems in the past. But luckily, we all made it both there and back safely. So now we can sort of laugh about it. So now I'll tell you about the location of the expedition. This is a heat map of the number of specimens of birds in collections for the entire Amazon basin. As you can see, areas with dark red are places with really good representation of a lot of specimens, and areas in blue have really poor representation of specimens. You can see outlined in this box here. And keep in mind, number of specimens, this is like all species. So the Amazon basin is one of the most species-rich places on Earth, especially for birds. So not only do you want just like one representative of a species, but getting that geographic sampling of all the species in a given area is what we need to do taxonomic research. So that's where we went, the Juruá River, specifically the Middle Juruá, which was the only tributary of the Amazon that Emilie never got to go to during her scientific career. So as a tribute, we sort of filled in the gap that she never got to go to. And this is our boat. We were on the boat for almost a month straight and slept in these really fun little hammocks with bug nets, just out in the open for a month straight. It was quite the adventure, all in very close quarters. So we got to know each other quite well by the end of the trip. Luckily, we had an amazing crew and ate extremely well because there was like a whole kitchenette on the boat itself, and we had a lovely cook with us, excuse me, Donna Anna, who prepared us these amazing like fish meals, like fresh caught out of the Amazon fish, like every day for lunch. So normally in an expedition, I like lose weight because you're running around the forest. This time that did not happen. This was our scientific sort of mobile laboratory on the second floor. We cordoned off the normal like patio area and took it over for prepping of bird specimens. This is our whole setup. So we had a really awesome view of the river and got to prep birds like every single day. It was really amazing. This is sort of the process of essentially scientific taxidermy. So we catch birds in nets, and then we do really detailed dissection of every individual to collect a lot of really important data, not just locality, but also the date, things like whether it was male or female, was it in reproductive condition or not, what was the molt like, did it look sick at all? Like sometimes we collect birds and they have like weird tumors, which can be used for disease research. So there's like all sorts of associated data with these specimens, which makes them like very data rich. And that's sort of what we do as a member of the LSU Museum of Natural Science. One of our goals and jobs is to contribute to growing bird collections. And it's as a result of that, we have one of the largest tissue collections of birds in the world. So tissue meaning modern specimens with genomic resources and all these like super data rich specimens. And it's like a big tradition here. And it arises, like the power of us to be able to study birds really arises from this really amazing deep and detailed collection. which you can use to study things like systematics. So that's how different species of birds are related to each other. The ecology, like what habitats different species are associated in, as well as evolution and taxonomy. So in total, our expedition was 24 days long, 24 days in the field. And it was an absolutely amazing team. This is Glaucia's term, not mine, but the dream team, she said. But I can definitely second that because it was just an amazing group of people to get to work with. And I would say lifelong friends. We still have a WhatsApp group that we remain active in and keep regular contact in anytime anyone has a big life update. Our two Brazilian net specialists, Bruna Costa and Bianca Mattiata, were just, their ability to run mist nets to catch birds was absolutely amazing. They'd set up like 80 mist nets in maybe half an hour, whereas I'd set up like one mist net in half an hour. As a result of that, they got the nickname Truck and Tractor because they were just absolute powerhouses setting up nets, which you can see here, and like hauling all that material around and extracting birds and things like that, which is really hard work, as you know. And they were also experts in collecting and preparing big specimens. Traditionally, like a lot of bird research has now started to focus on much smaller species. And as a result of that, these bigger birds are really poorly represented in collections, but are still really valuable. Like Hoatzin here is the one bird being prepped on the right, and that bird is like a monotypic family. So there's only one species in that family of bird, and we really have no idea what that bird is related to. Like, where did the Hoatzin come from? It's this bizarre, it looks like a dinosaur. And so by having these specimens, we can then study those types of questions. We also had two of the most amazing and just like wonderful people I've ever met in my life, but amazing professional skinners, professional taxidermists, Gigi Lima and Donna Schmidt. Donna in particular, as I think the one holding the machete in this photo, she has been going on LSU expeditions since the late 70s. This is with Van Remsen and Ted Parker in this photo. And on this expedition, she prepped her 11,000th bird. So absolutely amazing. And Gigi, I think at the time was in the 5,000 range, but is now approaching 8,000. So Gigi is the future Donna. And during this expedition, you know, many hours spent prepping in the prep room, they developed a really beautiful friendship, which was really fun to witness. And this is Gigi giving Donna a back rub because they were so sore from hunching over the table prepping specimens. But really, everyone did an amazing job. This is Jessie prepping with Donna and Gigi, and you know, taking all the data and the tissues. It's so much work. In total, they prepared 495 specimens just between the two of them, which was absolutely incredible. And this is Donna with some of her hard work, and they both prep beautiful specimens. So the more beautiful a specimen looks, the better prepared it is, the more scientifically useful it is, because it's easier to take data on color and size if the specimen is nicely prepared. This is just a nice view from the back of our boat with our LSU College of Science flag proudly waving. And here's a photo of the LSU contingent. My lab mate, Jessie Salter, as Glaucia said, recently defended her PhD and is now at the LA County Museum in Los Angeles. So if you get the chance, invite Jessie out for a talk as well. She's local. And then Marky Mushler is now an undergraduate like research associate with the Moore Lab of Zoology, also in LA. So just gonna plug my friends there. They're both excellent. And here we are working hard in the field, setting nets and bringing water to the guides. And Jessie here is prepping a bird. It was the first time she got to go on a big international expedition. I think it was a really wonderful experience for her as well, although very tiring. One of the things that made it tiring is the Girois is very muddy place. It has all these switchbacks.
Should have taken your boots off.
So this is Jessie extracting her boot from the mud.
I know. Thank you. I
And
appreciate
crawl
it.
out with sticks on like Spider-Man crawl because it was so marshy that you
Oh my God.
sink.
Woo!
I took this video of her from the shore. And then our, of course, fearless leader, Glaucia, eating, she's a very small person, so I don't think I've ever seen her eat as much as she did on this expedition, but it was really wonderful to get to go in the field with her. I'd always sort of looked up to her as my lab mate, older, more senior lab mate. And she absolutely crushed it as our expedition leader. So it was really, really fun to get to go with her and see her sort of in her element. And I think here she is eating some cake. I mentioned another really key expedition participant, Donna Ana, was our chef. Would make these amazing meals and cook us cakes in the afternoon, which kept not only Glaucia happy, but all of us very happy and well fed. Josue and Almir were our fearless boat drivers, boat captains, and guides. They, we hired them to really just drive the boat around, but they got really into it and came out in the field with us and helped set mist nets and were interested in the birds. So they were absolutely amazing and really fun to be around. Nice, very happy people. The biggest downer of the expedition, the expedition participant that we did not want but had to deal with, was all of the bugs. The Juruá River is famous for being, because it meanders, it's very like a windy river. There's a lot of standing water, which not only makes it muddy, like you saw in the video, but it means there's an absolute ton of insects everywhere. And I'm not exaggerating, this is what we had to deal with at night, especially around dusk. The bugs were absolutely crazy. So they would come in and swarm. Every hour, the type of bug that we were being swarmed by was a little bit different. There'd be like flocks, swarms, I guess, flocks is a bird term, swarms of bugs that would come and go, which made it kind of miserable. This is Jessie's hands after she'd been bitten by a bunch of gnats. This is what those gnats look like. And we affectionately gave her the nickname Miss Pium because she got the most gnat bites of anybody on the expedition. It looked like she had some weird disease every day because of the number of bug bites. Another bug we had to deal with is called poto, or the flamenguista, which is this insect with like a toxic chemical on its skin. This is not a photo from our trip, but this is kind of what it looks like. And it happened to Glaucia, where she was just like in the boat and this insect crawled across her skin and she totally broke out in this rash. So there's, the fun, beautiful part of expedition is the birds and the views, but there's also like big challenges. But
may I ask, Anna, I ask if anybody was collecting insects while you were there and being swarmed?
Not on purpose. I definitely had some in my prep kit that, got stuck. There were so many, they would just fall and die all over the place. So, but not on purpose. We did collect, one of the things we take is stomach contents, and those oftentimes have a huge number of insects in them. So sort of, I guess is my answer, but not explicitly the bugs that were attacking us.
Well, in the next trip, you can
yeah, definitely.
Some of them. No, that's awesome though. Sorry to interrupt, but I was just
you're good. I will talk about a few things that are not birds. So this is where we went, Caruarí, so this is the middle part of the Juruá. And there's all of our expedition points. So we went to I think 10 different localities. And as you can see, the river is super bendy. So it took a really long time to get from locality to locality, even though they don't look that far apart. The other thing you'll notice is that we tried to collect on both sides of the river, which I will bring up later because it's important biologically for the birds. We got to visit many of the amazing communities there, which were really welcoming. Their community is descended from people that moved in during the rubber boom. And one of the really fun things about going there is that we got to interact with local school kids, and Bruna and Bianca especially were leading several sort of outreach activities, talking with the kids, showing them bird guides, teaching them how to use binoculars, which we're really trying to promote conservation and that mentality, why should I care about the creatures around me? And in process is sort of a little mini field guide to the most common birds that we saw at all the sites we went, which we're hoping to send to the communities afterwards to keep this momentum going. Because it was one of the things that was really nice to see about this region is that the forest is actually really well conserved. And we want to, promote that and encourage people to keep protecting the forest. And the communities were like really excited to get engaged with this conservation mentality. So what did we find? The birds. Well, we documented, there's sort of couple main habitat types in the Amazon, and in the forest particularly, this is called Varzea. So this is seasonally flooded forest where the water during the rainy season, the bottom parts of the trees will be completely underwater. And that has its own specific community of birds in the Amazon, which we were lucky enough to get to see and collect samples from. On the left is the lemon-throated barbet, this really flashy, like the red heads is the male, and then scarlet-crowned barbet as well. Opal-crowned tanager with the amazing blue plumage and a very like iridescent head. This agami heron, which is a pretty poorly known bird, it's very secretive, was really abundant on the Juruá, in part probably because of all that muddy, flooded habitat. We collected some of these wire-tailed manakins, which you can see have these exaggerated tail feathers. The western striped manakin. Manakins are some of these birds you may have seen footage of, like the Michael Jackson dance. Well, that's manakins. We also saw sungrebe, which has these amazing tiger-striped feet, and the Purdus jacamar, which was one of the species we were interested in documenting for this trip in particular. There's like a difference between north and south. There's two different species and nobody knows why or where the distribution, the range of one species of jacamar ends and the other begins. So we found the species Persianus, which was sort of a new point on the map for that species. And I mentioned the Hoatzin. This is a better photo of it. This really weird dinosaur looking bird, as well as some cool parrots. And everybody's favorite, the tanagers and the cotingas. Just in, when you think of the Amazon, these are the birds you think of. The masked crimson tanager, plum-throated cotinga, and in these amazing flocks. We also got to visit habitat type, like River Edge has its own habitat type as well, which is more like sandy beaches, and things like cane will grow really thick. And there we collected horned screamer, Orinoco goose, both of which were first tissues for LSU. So this is the first time that we have this genomic resource to really study in depth what they're related to and sort of the adaptations present in the genome for these birds. We also collected some cool records like a dark-billed cuckoo, Lesson seed eater. This group of Spirophila is really poorly known. They all look the same. They all interbreed. It's like the taxonomy is very confusing. So that was a first tissue for LSU as well. And some cool range extensions of these river edge birds. So the Snethlage, the Amazonian black tyrant, and Spirophila americana, wing-barred seed eater were both pretty large range extensions. So that's important for, again, conserving the species. Like, you need to know where it lives and how big its range is before you can conserve it. And the last and perhaps most important habitat we surveyed was called terra firme. This is a forest that is in slightly higher elevation, like harder ground that's not seasonally flooded. The trees can be much taller in some cases, and it has its own distinct community of birds as well. So some of these were the green-billed toucanet, the Solenodera, everybody's favorite curl-crested aracari, which have this really unique head plumage where they have sort of fused barbs in the feathers of their head, and it looks like the tape from a tape cassette, if you remember those, like way back in the 90s. We also got musician wren, which is what you're hearing from this really beautiful melodic bird. You hear that in the forest, and it makes you think the Amazon and the rainforest. And antbirds in particular at LSU are really famous and have been studied for a long time. Glaucia's Glaucia is doing her dissertation on Ragmaturina, which is the bird in the upper left with this blue eye ring. And we were able to find some of those on our expedition. Many of these are obligate army ant following birds. So these big swarms of ants come through and the birds follow them and eat the prey that those ants scare up. And the other cool fact, this bird on the right with that white chest and sort of striped tail, the genus is called O'Neilliornis, named after the LSU ornithologist John O'Neill. And he's like really well known in the bird world. So it's pretty cool to get to collect birds named after, you know, people at your own expedition. And some not birds. You're asking about the insects. Well, I don't think we have any photos of insects, but this frog, for example, on the left was a poison frog only recently described in 2016. So this in general for all taxa, this part of the world was pretty poorly known. There's really cool iguana, really gigantic caiman, which Glaucia asked me to put in this this size reference, which is not completely accurate, but they were really, really big. And it was, we were not allowed to swim in the river, I'll say that. We saw lots of cool monkeys, caught the occasional bat, including this really amazing bald hukari, I think is how you pronounce it, as someone who doesn't study monkeys, with this amazing bright red face, which I think has to do with their breeding. My favorite non-bird thing that we saw, non-bird taxa, these are my photos, is the river dolphins. So there were not one, but two species of river dolphin. This is called the tuxi. And then we also saw the pink river dolphins, which have these like elongated snouts with teeth. And they would do this sort of like sparring behavior sometimes. No idea why, but it was absolutely amazing. They were all over the river at night, you know, in those hammocks on the boat. You'd fall asleep and hear the of their breath as they swam alongside the boat. So it was totally something out of a dream. There we go. We have two insect photos for you. Butterfly, grasshopper, no idea what species they are. I think some of these we've uploaded on iNaturalist, so maybe they'll get identified one day. But in general, the species, all species from this part of the world were pretty poorly known, including plants, these amazing like beach flowers and mushrooms, sort of like basket-like mushroom. And unfortunately, some skin mushrooms. This was me, like when you get your neck straps from backpacks, things rubbing doesn't only, you know, cause a callus like it might in drier places, but you also have sort of some mushroom forming problems, some fungus problems. So another with the unexpected expedition challenges.
You were colonized.
Yeah, I was definitely colonized. And then speaking of expedition challenges, the other thing that happened is, as you can imagine, you're on a boat and we're going through these big turns and it's really muddy. Well, it also got really shallow. So we dealt with that one day. This is a video of Jesse telling you what's going on.
Okay, we're on the boat. It's Saturday. We're having like sort of a leisurely cruise moment and we hit a shallow part of the river. And I feel like we're doing the equivalent of a 3-point turn right now in the boat. But it was, everybody on life jackets, stand in the middle of the boat was the word we just got from the boat driver. So, yep, hopefully we're not going for a swim today.
I think we're good.
So there you go. We did not end up going for a swim, but I think it was kind of a close call.
Okay, we're on.
Because everyone, the boat drivers that we saw running around and Marco and Glaucia, everyone looked like this. So that gave us a little bit of a fright, but it ended up being okay. So now I'll tell you about the scientific discoveries, which is the real reason we were there, not just watching river dolphins. So in the Amazon forest, rivers delineate species, and many taxa of birds, when you cross from one bank to another, it's a closely related species that exists there. So they sort of allopatrically replace each other. And this is a map, a good example of the Ragmentarina antbirds that Glaucia studies. You can see between rivers is a different species in sort of this wheel-like fashion. One cool thing about the Juruá is, as I've mentioned a couple times, it's very bendy. And because of that, chunks of the river, if you watch this centerpiece, I'll start it over again. This piece right here goes from the right bank, slowly gets absorbed, and then becomes sort of like an oxbow lake on the left bank of the river. So you have this land switching banks. And because of that, it was thought for a long time that maybe this pattern of species replacing around rivers might not matter for the Juruá, because they would replace, but then you have the river changing sides. And so birds could maybe cross the river that way, even if they wouldn't normally fly across open water. But when we went on our expedition, we actually found the opposite. So this is a photo of Lepithyrix coronata. And what you can see is on one bank of the river, the birds were bright green, and on the other bank, they were black. And this held for the whole expedition, both sides of the river. They were green on one side and sort of this blackish blue on the other. We found this not only with Lepithyrix, but also Epinacrophila, the stipple-throated ant wren. The right and left bank look different, with birds in the left bank having that darker throat. And same thing, the females look different, with a little bit more orange in the throat on the right bank. This is the common scale-backed antbird in the genus Willisornis. And again, you can see one side on the left bank, they have this dark throat, and on the right side, there's nothing. And one cool thing about antbirds is that it's oftentimes the females are the more pronounced geographic variation. So in the females on the left bank, they're this totally orange color, and on the right bank, they're all gray. So it was even more pronounced in female birds. And our most exciting discovery, this is Mimoborus myothrinus, the black-faced antbird. On the right bank, they're this total orange color, and on the left bank, they're completely white. And when we took these birds back to the collection to compare them, there's no current subspecies, no taxonomic name given to the left bank birds. So we're actively studying this, trying to find out if this group of antbirds might be a new species, or at least a new subspecies to science. So at the end of all that, 21 days in the field, we collected 913 specimens, over 3,000 tissues with really high quality genomic data. We recorded 423 species and collected 221 of them. So we did a really amazing job. This is Glaucia with the entire series back at our collaborating institution in Sao Paulo. We also submitted much of this data to eBird. As you might imagine, like if you look at the eBird hotspot map, this region was like totally poorly known before we got there. So we uploaded all this data to eBird, and hopefully that will help eBird create better filters. And as the filters get better, that encourages more people to go bird there, because they know what they're going to find and they have a better sense of what species occur in that area. And we recently were able to publish all of this data in a scientific article in the Journal of Ornithology, which, fun fact, was the journal that Emilie Snethlage used to publish in Germany. So we sort of, partly as tribute to her, wanted to publish in that same journal, documenting these changes on the left and right bank in different taxa across the Juruá. And the reason we the hypothesis we came up with to explain that is that even though the river changes bank, it's still staying in this really wide band of Varzea forest. So it's still flooded, which means that the terra firme, that upland drier forest, never comes into contact. So the birds that we found this change on were always that terra firme, higher elevation, like associated with that habitat type. And one other really awesome aspect of this expedition was that we had a documentarian on it, Angela Portillo, who's Brazilian American and has previously worked with BBC and a couple other groups. She came, and here she is filming Glaucia fake cutting a trail. And yeah, so she was a really integral part of the team, and we're really looking forward to see the footage as it continues to come out in the next few years. But this is the whole group taking a selfie in the tip of the boat. We also, have to give a shout out to Marco Rego, who is Glaucia's husband and was also the drone pilot for the expedition. So Angela needed like an assistant to help her get more footage of the expedition, and Marco came along to make that happen. and he got to play with the drone the whole time. So I think was very excited about that. This is some of the footage that Marco got. So he was really excellent at his job. And now this is this is Glaucia's poster in advance of our documentary, but we have a teaser trailer here for you today. So I will play that for you.
Meu Deus do céu.
So there you go. And with that, I'd like to thank everyone that made this trip happen, many funding agencies and the people doing the paperwork behind the scenes were pretty integral to this whole expedition. And with that, I will take any questions.
Thank you, Anna. Thank you, and thank you, Glaucia, who's in the wings there, maybe somewhere.
Yeah, I'll pass her your message.
No, that was great. Thank you for the overview of this great project. I know there are a couple questions that came up in the chat, and I'm sure other people have questions too, but I'd like to ask a couple just to kind of start the ball rolling if you're up for it.
Yeah,
And
sure.
congratulations on getting through this, because expeditions are challenging. I've done a few too, and you know they do push us physically and emotionally and mentally. But that of course is a big part of the reason why we do them, because they do push us out of our comfort zone, and by doing that we learn so much, you know, just by being there in the field and really seeing the conditions and getting a true on the ground sense of where these birds are living and what the conditions are for them. You know, we gain so much more understanding about their ecology. So my first question is, you know, you said that this particular river was not sampled by Dr. Snethlage, if I'm saying it correctly. I'll say it the German way. So it wasn't sampled by her, but there were a lot of other areas that were sampled by her in the early 1900s. So first off, I did wonder how much of her, how many of her areas that were sampled in the past have been resampled separately from your expedition? Have there been also additional expeditions to do a resurvey effort, kind of like they're doing with Grinnell's work in California?
Yeah, that's a really good question. In terms of resurveys specifically, I can only think of 2 examples in the entire Western Hemisphere tropics that have done resurveys, and that's there was Ben Freeman and colleagues from Cornell, John Fitzpatrick, redid a survey in Peru. They weren't collecting, they were just doing MISNet data to try and see, it was called the Elevator to Extinction. You might have seen it, it sort of went very big on New York Times, to look at whether distributions of birds had shifted, but in a much shorter period of time. It was only like 30 or 40 years. And they found that some things had shifted, but it's also really hard for resurveys in the tropics. We still have such poor baseline data, and our trip is a good example of that. Like to do a resurvey, you have to really, really well know like how much natural fluctuation is there in the tropics. Do things just occasionally go extinct because there's so many species of birds, and maybe some sort of weird weather pattern happened just once, and it, you know, naturally a couple things went extinct sort of locally, were not there for a period of time, and then naturally would recolonize it in a healthy forest. So there's that Peru expedition, and then in Colombia they recently have been doing Alas y Cantos y Colores is the name of that sort of expedition working group, led by Daniel Cadena and Andres Cuervo. out of the Humboldt Institution in Columbia, doing more museum resurveys and collecting, but much more focused on anthropogenic impacts. So going to places and looking at the forest, whether it's been cut down, how that changes whole bird communities, and what sort of ecosystem services have been lost. So in the Amazon, to my knowledge, there has not been a big resurvey effort in the Amazon. And in part, that's just because we don't, to this day, we don't even know it's there. You really need like really good data in order to do a resurvey. But I think there's also increasing interest to try and make it happen anyways and figure out ways to maybe simulate some data, to try and make resurveys happen. Because what's happening in the tropics with climate change and everything, it's a really important question that a lot of people are.
I imagine part of the difficulty also of resurvey efforts in the Amazon basin just has to do with the ferocity of the Amazon itself. Because I work on river systems and I know how much it can scour from one season to the next. So just the river itself, the dynamism of the river can change, you know, what you're seeing bird-wise, as you're saying, and just as you pointed out. So, okay, so thank you. I was wondering about that. And then in regards in particular to your study though, to your expedition, what is the width of the river?
Yes.
Oh, that's a good question. Off the top of my head, I don't know, but it was only like, in some parts, maybe 200 feet wide. It's very narrow, which is why people thought it wasn't a barrier. If you know much about the Amazon, in parts of the Amazon, it can be, you can't see across the river. It's huge. And that's why it's a barrier to birds, especially small understory birds like antbirds. They don't like to fly very far, especially in open habitat. They're used to dark understory where there's lots of cover. So they almost never fly across rivers, and that causes a barrier, which then things genetically diverge with time, and that's what causes these species to be delimited by rivers. The Jurawa is super narrow, so everyone just sort of assumed it wasn't a barrier. And then you combine that with this very dynamic river flow, and it was sort of crazy that we found what we did. And we really, again, it has to do with just that amount of flooded forest.
And the fact that they were basically in like refugia and the terraces above the river. It sounds like it's fascinating, so interesting, and so worthwhile to go and sample it, you know, because instead of just assuming that you knew what would show up, you know, thank goodness you were also working away from the specific river corridor, and you actually went out in some kind of band into that more static vegetation on the side. Okay, and then Howard had a question. I think there are some others too, and I'm going to answer or ask Howard's, and then other folks can unmute themselves and ask too. So Howard asked, how did you decide on your sample locations and your sample points along the river?
So it was a couple of different things. The decision to go to Karawari was in part to start to do the middle Jurawa, was in part driven by the fact that there's an airport. So expeditions, a lot of this is just like logistics. And, you know, the sample points that we hit, it was partially like, how far can we go in a month? Because as I said, boating all of those curves, we didn't go that far by air, but it was very long days to go through some of these meanders on the boat. And then the specific sites we picked, we wanted to target that upland forest because we knew it was such an interesting habitat type. So we picked areas, and from Google Earth, you can actually zoom in and see, and there's, you know, really nice satellite like topography data now. So you can oftentimes sort of predict where that's going to be. Glaucia also had friends that had done more like consulting jobs and sort of basic survey work, not collecting in this region, who she was able to talk to, and they sent her points that they suggested we visit. And then some of the places we also would just like pull up and be like, cool, we can park the boat there. That's accessible, you know? Or is there a community there whose trails we can use? Because especially on the right bank, you'll notice in our figures for that paper, there's a really big sample difference between the left bank and the right bank. That's because it was impossible to find really good upland forest on the right bank. It was really, really inaccessible. The one place that we were able to get into was a community, and the community actually wasn't on the edge of the river. They were up a small tributary and had a sort of like fire road trail kind of thing. So in the wet season, they take boats up to their community. In the dry season, they hike. And we were able to use their trail system to go way farther in than we would have if we had had to cut that entire trail ourselves. And so because of that, we like knew that this community existed, and so we wanted to go there because it was more accessible. So some of it was just logistics, but then also trying to target that habitat type.
Well, that totally figures. I mean, logistics drive the expeditions ultimately, you know, I mean, they really do. And poor logistics can ruin an expedition. So, and lack of emergency medical training. So that was really nice and good thinking that you did the training too initially. Does anybody else have a question, Dawn?
I have a couple of follow-up questions now. First of all, thank you, Elena, for asking about the width. I want to follow up on that. That was puzzling me because I was surprised by how you get evolutionary differentiation with what appeared to be a fairly narrow river, which doesn't act like much of a barrier. So you're suggesting that it's the flooding, and then the follow-up question is how wide an area is that flooding?
I think it's much, I don't remember, I have to go look at the paper to get the exact measurements. So from satellite, we could get a little bit of roughly how wide is, I think it's, I mean, in parts from hiking it on the right bank, like that site, it took us an hour to hike in. I think it was maybe 10 kilometers from the river, and then there's the other side of the river. So in parts as far apart as 20 miles maybe.
Okay, so it is quite wide.
It's quite wide, but it just doesn't look like it on satellite. Yeah.
So for those birds that whose habitat does not include flooded marshy areas, then they're driven back.
Yep.
And so there is really a wide environmental barrier there, even though it doesn't appear to be.
Yeah.
Okay, so that makes sense. And I guess the only other observation, I did notice the difference in number of species in your photograph, and I was going to ask if the left bank, right bank ends were really different. And I guess you kind of answered that. It's kind of just a logistical thing. But why was one bank more accessible than the other? Is that just because of communities being there? And
it was also the presence of the terra firme. So for whatever reason, on one side, there was just more terra firme close to the river than on the other side. And that also might play, and it could be a historical thing, like Linnea was saying, rivers are very dynamic. So it could have been that in the past it was closer to, and we also went during the dry season, because you can imagine working the wet season would be awful. Again, logistics.
Sounded awful regardless.
It was fun. We like to play up the drama for these talks. It really was amazing. So you get past the bug bites, but in the wet season, the terra firme is much more accessible because you can take boats up these small tributaries. We couldn't because they were so dry. A lot of them were really hard to get up. So it was partially like our own doing, but it would have been way worse to go in the wet season. So we were happy hiking in, even if that meant, we got fewer birds, but.
So I guess the only other comment then is it's not a random sampling. It really is kind of biased towards accessibility. So that has to be taken into account.
Yeah, especially if you were to do like population genetics on these birds, you definitely have to take sample size into account. You can do things like randomly drop a few individuals from both sides and sort of simulate what would happen if I had fewer or more data. So we do have to take that into account. But even with our pretty small sample size, I mean, you can start to pick up pretty clear differences. And we're also hoping to go back. So I was telling Linnea this at the beginning when we were sort of setting up the Zoom, that Glaucia recently got a job as curator of birds at the Florida Museum of Natural History, which comes with startup money. So she has funding for expeditions. And we're planning to go back in 2024 to the upper Juruá. So as you go, it's actually south, but if you go upriver, away from the Amazon is upriver. So the Amazon is north of the Juruá. So we go south, upriver, and the river gets narrower and narrower. So the follow-up question to our first publication is, well, what happens when the river and the Várzea gets narrower and narrower? How narrow does it have to be when you lose that pattern?
Yeah.
So one thing that both Lausia and I studied for our like actual dissertations, this was just a side project, is what happens when birds come into secondary contact. So two species formerly isolated by like a river meet. Do they hybridize? Do they not? Do they only hybridize a little bit? Do they hybridize a lot? What do hybrids look like? How wide does the river have, how narrow or wide does the river have to be for things to hybridize or not hybridize?
And how long have they really been separated?
Yeah. So we hope to go back, get more samples to partially solve that sample size problem, but also to figure out, yeah, what happens upriver.
Yeah. Interesting.
I'll mention one thing about sample size, so sample size when you're doing an experiment, sample sizes when you're doing kind of repeated measures in a particular place is different. And those kinds of projects are different from an expedition in the sense that an expedition is usually like a first time.
Yeah.
Or maybe a second or third time, but it's usually just, it's being mounted because it's really challenging to get to this location. It hasn't been sampled repeatedly before. And so people are going in and just kind of collecting baseline information. And usually, you're getting enough information that you're going to be kind of tantalized and you're going to want to go back. And then you can start to set up more logistical structure, so that you could go back repeatedly and such, maybe in one of the local communities, you could have a location where you have a village house that you stay in repeatedly, or there's some kind of support when you go where you could do more intensive sampling. So I see this as like, it's hard to worry about sample size when you're just kind of going in as a pioneer.
My comment was before this n was equal to 0. So
right, exactly. So anything is good.
Yeah, everything is good.
Yeah.
It's also partially why we collect birds instead of just observing them, because when the hole is that big, you really need data rich specimens and that material to go back and revisit. So now, like, those specimens, when we collect tissues, we collect an absolute ton. So people can go and resample those and use them for a ton of different projects. So that's
as well as then conduct population estimation studies and behavioral studies and habitat use studies and everything else that would kind of come off of those. So okay, so there's another question, and this is for you personally. Anna, this is from Randall Searcy. He said, thank you for dedicating your life to exceptionally meaningful work. He's wondering about your backstory and that critical period in your life when you fell in love with nature and decided to pursue it, as a profession, the study of it as a profession.
Yeah, well, thanks. That's such a nice statement. You make me feel very important.
You are.
Yeah, so I grew up in California, actually. I grew up in Davis, you know where that is. And I grew up camping all over California. Every summer, my family, we do like a family reunion. And instead of going to like a hotel or doing, like a potluck, we sort of like normal people things, we go camping. Even with my 90-year-old great aunt still goes camping at 90. So I grew up running around Joshua Tree and Big Trees and Yosemite and Tahoe. And yeah, so to me, like going on a boat for a month in the jungle is like, cool, that's summer break, I also grew up really traveling a lot. My family especially would go, like, my grandparents were from San Diego, so we'd like drive to San Diego and then drive to Mexico for the summers and go see birds. I saw my first toucan, I think at age 8 in the Yucatan in Mexico. And in part, my mom is a lawyer, but she worked for Fish and Wildlife. So I call her like a hippie lawyer. So she, I also would grow up and go to like take your kid to work days to Fish and Game and get to see desert tortoise shells that were confiscated by US Fish and Wildlife and things like that. So that was sort of where the love of nature was really instilled in me. And then my real like taxonomic preference for birds, I can say was a result of my undergraduate education. I started at Berkeley as a freshman and got really interested in research, and I really wanted to work in a lab, and I contacted this guy who was a grad student working on lizards in, I think, Indonesia and oil palm. I was super excited, and he ended up having me do nothing but enter data in Excel spreadsheets all semester. And I was like, yuck, this is not what I want to be doing. So I took a break from research, and then a year later started up again as a sophomore and contacted the Museum of Vertebrate Zoology about volunteering opportunities. And I ended up doing bone numbering, which for those of you familiar with museums, you'll know what that is. But basically every skeleton gets the museum number written on it in rapidograph, like special archival ink. So that's what I was doing. And I absolutely loved it. I could sit there for hours, like trying to number the bones of a hummingbird, which are really tiny, and getting to see like, what does a turtle skeleton look like? All those sorts of things. And so from that on, that was in 2011. I've worked in museums ever since. I started working for Carla Cicero, who's the staff curator of ornithology at Berkeley, and ended up working, by the end, I was working 30 hours a week and only in class like 4 hours a week as a student, working in the bird collection, curating all that material. I got to go on my first expeditions there as an undergrad, and it was totally amazing. You asked about the Grinnell resurvey, like I got to actually go on one of those trips as an undergrad, meeting Jim Patton, the mammologist you might know, who, fun fact, went to the Juruá, spent a year collecting mammals in the Juruá. He and one of his Brazilian PhD students, she's now, I think, an equally famous Brazilian mammologist, collected mammals all up and down the Juruá. There's a photo of Jim's boat falling over in that monograph. So those were the people I was really inspired by. And they also just like gave me a chance. This, undergrad is really enthusiastic, won't go away, and let's support her and encourage her. So
that's awesome. You certainly were immersed in it from the time you were young, and that helps a lot too. You were just surrounded by it. That's very awesome. So I'll ask if there are any other questions at this time from the folks who are here. Does anybody else have any comments or questions? Renee, what do you think about the number of specimens that were?
Yeah, no, that's great. I was thinking like that was a breeding season, right?
So we don't really know. Tropical birds, the breeding time of tropical birds is like super poorly known. One thought is that tropical birds, because there's way less seasonality, like here it's, winter and spring and summer are like very distinct seasons. In the tropics, it tends to be more, there's like a rainy season and a dry season, but there's not, that's sort of like 2 seasons, right? Instead of 4. So one theory is that birds have really long windows of reproduction. They can sort of reproduce at any time. And it's a lot less synchronized. So different species will breed at different times. And we don't really have a good understanding of that yet. They tend to breed, I think, right before the wet season. So we were there in July and August, in August actually. And that would have been a little bit before breeding really, really started, but just at the very beginning is when we were there.
Okay, yeah, because I kind of jealous. I mean, when I see you there, because I used to do it in the 80s, and now it's for a long time I haven't been there in the Amazon, but it's amazing. Yes, and all of those bugs and all of that is worth it. And so yeah, it's worth it for that, to being in there. And so yeah, that was a great talk, and thank you for sharing with us.
Yeah, thanks so much for having me. This was really wonderful.
And of course, there's a lot more information that can be gleaned from the specimens that you collected that, you've only just scratched the surface with your publications. But those specimens now are there for other students to work on, or for you to continue working on into the future. I assume there were other graduate students who are going to use some of these specimens as the basis of their work too. But are there other papers that you're working on right now?
Yeah, so I know that there's a bunch of other students using the specimens, not necessarily that I'm like personally involved in, or even Glaucia. Like the Lepidothrix, that mannequin with the green black, there's 2 different students studying that right now, one doing the genetics and one looking at feather microstructure to figure out why, like what structures are involved in that color change. And then in terms of our like specific expedition group, the next big paper we're going to do is that taxonomic study on the Myomovirus myothorinus and looking at the females and describing that taxa that currently, as far as we can tell, does not have a name associated with it. So doing which is like part of, it's like a dream of any museum ornithologist to get to describe a taxa new to science. So that will be the next like really exciting publication.
Of course, very exciting part. And so when do you defend your dissertation? Are you writing right now?
I am writing. I'm writing my first chapter and finishing the lab work for my second and third chapters. I was one of the waylaid by COVID grad students, and luckily LSU is very generous with timelines and extensions. So I have a little bit less than 2 years left.
Okay.
In part, I'm actually planning to do another expedition as a 7th-year grad student, which you're not really supposed to do, but again, COVID like waylaid everything. So I'm hoping to go to Bolivia for 2 months in December and January to study another hybrid zone that's actually for my dissertation. So I study the Diglossa flower piercers, if you've ever heard of those. It's a type of tanager, which are famous for their hooked bills and nectar robbing behavior. Which is not directly what I study, but they have hybrid zones. So I use them to study, again, that sort of speciation dynamics. So I'm going there in December. And then once I do that, then I'll graduate.
Well, you're going to have, of course, for many of these projects, you're going to have data to work on for years.
Yeah.
And you're gonna have, a whole long professional career ahead of you publishing on these. So I know I probably speak on behalf of others, but congratulations on your work and good luck to you in the preparation of your dissertation, your other expedition. And thank you so much for sharing this expedition with us. It's awesome. And if anybody else has a last question, please feel free right now. Otherwise, I will close the meeting. We have a lot of thank yous in the chat session, by the way. Thank you, Anna.
Thanks, you guys.
Looking forward to Expedition 2.
Yeah, definitely.
And the movie, that was a very thrilling, teaser.
I know the music is quite dramatic.
And I love the Avengers, look at the beginning.
That was Glaucia. She has much of this talk. She made the slides for us. That was all Glaucia's artwork. But I will end and just say that for my dissertation, my first chapter, actually, I included some samples that I got from the Western Foundation in my first chapter. So, every museum, every specimen is super important. You never know what it's going to be used for. The old, they were like really old specimens too. And those keep on giving. I got genetic data out of them and for the flower piercers. So thanks to you guys for, maintaining your collection and keeping people excited about museums.
Thank you. These specimens, every one matters so much. So they have to be really, really carefully taken care of. So, all right. Thank you so much.
Yeah.
Anna, thank you. We appreciate your time, and please say hello again to Glaucia and thank you to her too.
Yeah, I will.
That you use the presentation. So
Thank you, Anna.
Yeah, thank you guys so much.
all right.
Thanks. Nice meeting you.
Thank you. Thank you so much.
All right. Take care, everybody.
Bye.
Bye. Wonderful
Bye.
presentation.
Bye.